Hello everyone, this is Rohit Shankar and this is our fifth lecture of pure culture. Now in this lecture we are going to understand Urease and ONPG test for the identification of unknown bacterial culture. Let's start our lecture. Now first test is the Urease test. Now see what is the purpose of Urease test. The first purpose is to determine the ability of an organism to produce the enzyme urease which hydrolyzes the urea. To identify the rapid urease producers such as Proteus and Morganella and weak urease producers, Klebsiella pneumoniae and species of Enterobacter. Now what is the principle? Urease splits the urea molecules into ammonia, CO2 and water. Ammonia reacts with the solution to form an alkaline compound, ammonium carbonate, which results in an increased pH of the medium and color change in the indicator to pink red. Now see, first what's happen, let's see the reaction. Now see here, what is the principle? Now urea reacts with the water. In the presence of urease, urea converted into the CO2 H2O and ammonia. Then further ammonia converts into the ammonium carbonate. Now due to this ammonium carbonate, there is a production of color. There is a color change in the reaction, so which indicates that there is a production of urease by the unknown bacterial cell. What is the interpretation? Let's see. If you have to have Christensen's agar for the test, and if you got positive result, then we are going to say that there is a rapid urease activity rate throughout the medium. Positive, slow urease activity rate in the slant, initially gradually converting the entire tube. Negative results, there is no urease activity, medium remains yellow. Now see, these are the results. Now positive results for Proteus species, color change. B, Klebsiella pneumoniae, positive results, there is slightly color change. C, negative for Escherichia coli, there is no any color change. Next is ONPG test. Now what is the purpose of this test? To determine the presence of late or slow fermenting strains. To distinguish some Escherichia coli, Citrobacter species and Arizona subspecies, that is ONPG positive, from similar Salmonella subspecies, that is ONPG negative. To speciate Shigella, since Shigella soni is the only ONPG positive Shigella species. Now what is the principle? Two enzymes required for lactose fermentation, lactose permease and beta-galactosidase. Now what is the function of lactose permease? The activity transfers lactose into bacterial cell. Now what is the function of lactose permease? The lactose permease gives permission for the lactose to enter inside the cell. Beta-galactosidase, it degrades the lactose into glucose and galactose. Next one, lactose fermenters. These possess both enzymes. Slow or late lactose fermenters, there is no permease activity, only beta-galactose activity is there. Non-lactose fermenters, there is a lack of both enzymes. What is the interpretation? Positive test, it gives us yellow color within 20 minutes or to 24 hours. If we got negative results, then there is no color change or colorless after 24 hours. We can observe this test. Now in the first test tube, there is no color change. In the second test, there is a color change. So first test is a negative for Salmonella typhimurium. Second test is a positive for the Escherichia coli, which is ONPG positive. So this is all about these two tests which are used for the identification of unknown bacterial culture. Thanks a lot.